Well, the design of this phase 2 trial is very unique in that the patients are highly and deeply phenotyped. We're using high-tech imaging modalities like MRI, we're using actigraphy, we're using the CardioMEMS device. These patients are going to be extremely well phenotyped. And what does a well phenotyped population mean? That means that we can extract a lot of data from this phase 2 trial to design a really great phase 3 trial. And if we use the CardioMEMS in the phase 3, then we could even think about adaptive trial designs or Bayesian designs. It would be a very unique way to design that trial. But again, looking at messages or learning paradigms from this trial, to me, guiding therapy with the inherent knowledge of the pressures and potentially with stroke volume that we might be able to glean from the CardioMEMS design really gives physicians an opportunity to participate in personalized medicine for these patients. Pressures are the first things that changes in these patients. The late manifestations of those pressure changes and how it works on the right ventricle are reflected in later things that we assess, their functional class, their 6-minute walk test, some of the biomarkers, their effect on other end organs. Now we don't have to wait for that to happen. We know what the pressures are doing in real time using the CardioMEMS device, and we can act on that. And I think that's really important because as we evolve our paradigm for treating patients, right now we treat patients to try to achieve a low risk. That means a low risk of mortality and morbidity in 1 year. But we know that that prediction is based on what the right ventricle is doing. Now we know the afterload, and we know it soon. So we can act on that and personalize medications to that effect. And even in some cases, perhaps withdraw medications safely. And that's the important part. You want to do it safely. If we had a patient on therapy and we didn't think a therapeutic was working, we don't know. Maybe it's just keeping the patient there. But we wouldn't know that until a change in the 6-minute walk test happened way down the line, or a change in functional class, or if we brought the patient back to the cath lab in 8 to 12 weeks to see the effect. Now we'll see the effect of the pressures immediately, before the patient even knows something is going on. So the safety factor that brings when we are personalizing medication to a patient is going to be really unique. And I think that's one of the big learning lessons from this trial that we'll be able to extend to patient treatment in the future.